Hello everyone. In previous lecture we have discussed the role of ribosomes and its structure and its functions. Today we will discuss the prime function of ribosome is to protein synthesis. How protein synthesis occurs and what is the importance and significance of protein synthesis inside the body. So this is the structure through which we can understand the process of protein synthesis in form of transcription and translation. This is nucleus inside the cell. In the nucleus there is presence of genetic material. Genetic material means DNA. DNA is consist of original information in coded form, so it copies the information to the mRNA. This process is known as transcription, and mRNA reaches to the ribosome. Then it translates the information in form of synthesis. So the whole process from DNA to making protein is known as protein synthesis. DNA to RNA, RNA to protein is also known as central dogma of biology. So before going in the depth, we should try to know the basic structure of protein synthesis. This is the structure and this is the process of protein synthesis. Now let us take a look on protein synthesis, the blueprint of life. Proteins are, as we all know, that those are building blocks of life, playing an important role in constructing, repairing cellular structures, regulating biological processes, and determining an organism's unique traits. Because we are unique in itself. Protein synthesis is the process where cell makes proteins by following instructions from genetic material, that is DNA. It is highly regulated and essential biological process that ensures the proper functioning of cells, tissue and organs. Now next is understanding how protein synthesis occurs. Protein synthesis occurs in two ways, in two steps, that is transcription and translation in protein synthesis. These convert genetic information stored in DNA into functional proteins. Next is transfer of genetic information from DNA to a messenger RNA molecules by the process of transcription. So this is the first step in protein synthesis, that is transcription. Second step is translation. Through the translation process of reading the mRNA, means messenger RNA, messenger ribonucleic acid, sequence to assemble amino acids into protein is known as translation. So as I already stated, the process of transcription and translation. So from nucleus, there is DNA inside the nucleus, is copied in form of messenger RNA, and messenger RNA this process is known as transcription. Transcription is the process of information DNA to messenger RNA. This is transcription, and mRNA reaches to the ribosome. To reading the sequence in ribosome is into amino acids. The process of copying original information from the DNA reaches to the mRNA. This process is called transcription, and from RNA to ribosomes, where the information has been read out into collecting the amino acids in form of protein chain. This is known as translation. First is nucleus to reaches the ribosome is known as transcription, and from ribosome to making final product protein is known as translation. Let us take an example, muscle growth and repair. When we exercise, muscle cell experience small tears. The body responds by synthesizing new muscle protein, leading to muscle repair and growth. Second step is transcription. So those were the two steps, mainly transcription and translation. Now we have a detailed look to the transcription, how this process occurs into various steps inside the transcription. From DNA to RNA. Transcription is the major step in protein synthesis in which from DNA to mRNA we can understand. The first step in protein synthesis is this, means transcription, which occurs in the nucleus of eukaryotic cells and in the cytoplasm of prokaryotic cells. Now the important thing is that there are three main steps in the transcription, which are initiation, elongation and termination. In initiation, the enzyme RNA polymerase binds to the DNA at a specific region called the promoter. Second is elongation. RNA polymerase reads the DNA strands and creates a complementary mRNA molecules. Third is termination. The RNA polymerase stops transcription when it reaches a stop signal and the newly formed mRNA detaches. For example, hormone production. The pancreas transcribes insulin genes to produce insulin mRNA, which is later translated into insulin protein to regulate blood sugar. Now, second process is translation. In translation, there is converting of mRNA into protein. Once the mRNA is made, it moves to the ribosome. When the protein is built through translation, translation converts the genetic code into a specific sequence of amino acids which link together to form proteins. There are also three steps in translation like the transcription. First is initiation. In initiation, the ribosome binds to mRNA at the start codon, that is AUG. Second is elongation. Transfer RNA molecules bring amino acid to the ribosome where they are joined by peptide chain. So this is peptide chain. Ribosomes brings amino acids, means transfer molecule tRNA, transfer ribonucleic acid molecules brings amino acid to the ribosome where they join together by a peptide bond. Termination, this is the final step of protein making where the ribosome reads a stop signal in the mRNA and release the finished protein. Example is enzyme production. Cells translate DNA instruction to produce enzymes like amylase, which helps break down starch in food. Now the role of ribosome in protein synthesis. Role of ribosome in protein synthesis is proteins are assembled by molecular machines. Those are ribosomes. They can be found floating freely in the cytoplasm or attached to the endoplasmic reticulum. So the ribosomes, which are free, participate within the cell's function, whether rough endoplasmic reticulum made the proteins such as hormones and digestive enzymes. Now let us take an example, antibody production, in which white blood cells synthesize antibodies using ribosomes to fight infections. Next is post-translation modification. How the post-translation modification occurs. After translation, proteins often undergo modification to become fully functional. These changes may include folding. Folding is the first step in post-translation modifications in which proteins take their functional 3D shape with the help of chaperone proteins. Next is cleavage. In cleavage, some proteins require sections to be cut off to become active. Next is addition to chemical groups. Phosphate, carbohydrates or lipid groups may be added to enhance function. For example, hemoglobin maturation. In this, hemoglobin, the oxygen carrying protein in red blood cells, undergoes modification before it can efficiently transport oxygen. Next step is Protein synthesis and genetic traits. Protein synthesis determines the unique traits of organisms because protein controls cellular structure and function. Those are: eye color is influenced by melanin proteins, hair texture is determined by keratin proteins, height and muscles mass are affected by growth and structural proteins. For example, melanin and skin color. Cell produce different amount of melanin protein leading to various in skin tone because we all are different in our skin tone due to this. Next is error in protein synthesis and diseases. If any type of error occur in protein synthesis, what will happen? So various types of genetic disorder and diseases may be developed due to error in protein synthesis. Like sickle cell anemia is a burning example of error in protein synthesis in which mutation in the hemoglobin gene leads to the abnormal red blood cells. Next is cystic fibrosis. A protein misfolding issues affects lung and digestive system function. Next is cancer. Uncontrolled protein production can lead to tumor growth. For example, as we have already taken sickle cell anemia. In this, a single DNA mutation in the hemoglobin changes the protein shape, causing the red blood cells to become sickle shaped. Next is importance of protein synthesis in medicine and biotechnology. We would try to know and try to understand protein synthesis has led to advancement in medicine, genetic engineering and biotechnology. In the field of insulin production, genetically modified bacteria produce insulin for diabetes treatment. Gene therapy, scientists modify faulty genes to correct protein production defects. Vaccine development, protein synthesis is used to create vaccine components such as viral spike proteins in COVID-19 vaccines. Thank you so much.